Yes, dear 9th class students, welcome back to our NEET foundation classes. So it has been 2 weeks. We have not went with the class. So again, we will repeat the lecture 3, page number 25. If you have the manual, you can go with that, or else please go with your notes which you have been written previously. Okay, so fundamental unit of life. So here we are discussing about cell cycle. So you all members know that cell is the basic fundamental unit of any living organism. Okay, to continue our generation, it is the thing that we have to regenerate the cells. So in this, that is what it is, a continuous process. The division of cells is the continuation process. That is why the name it is known as cell cycle. So here, cell cycle, it is mainly taking place of two divisions. Okay, cell cycle, it undergoes with two divisions, that is mitosis and meiosis. Okay, mitosis and meiosis. So, in the previous class, we have discussed about what is that cell theory. Okay, this was proposed by two scientists, Schleiden and Schwann. It was proposed by two scientists, Schleiden and Schwann, and they have mentioned two statements. What the first statement? Okay, you have already studied in the general syllabus, but here it is also again repeated. And also in your 10th class, in the 10th class, you'll be having most of the part. Okay, half of the lesson, reproduction lesson, it is covered with total of this topic only, cell cycle. Okay, so Schleiden and Schwann, one is botanist and one is zoologist. They two members proposed a theory that is known as cell theory. So two main points they have mentioned. They postulated two main points. What is that? All living organisms are comprised of cells only. For example, I am and you. Okay, every living organism, what it is made up of? What is made up of? It is made up of cells. Okay, for example, if I say this is a chalk piece, what it is made up of? Chalk powder. From top to bottom, it is comprised of what? Chalk powder. Like that, each and every living organism is comprised of what? Cells. So the second point is what? All cells come from pre-existing cells. Okay. For example, if you consider this as a cell, this is the new cell. Okay. This is new cell. Where this new cell has formed from the old cell. Old cell means what? Already it is existed before this new cell. Okay. How we can say old and new? It is pre. It is post. Before, after. So like that, all cells, all new cells exist from the pre-existing cells. Okay, we are saying that living organ. For example, when we are at the age of what is that 0 month? Okay, we are very small, but after that we are gaining weight and we are becoming very height. So why? Because of the addition of new cells. New cells, where does they come from? Already existing cells are present now, that they will divide and divide. Okay, Once again, I am repeating. Okay, once again, I am repeating. For example, during our 0 month, we will be like 2 kg. Okay, we will be like 2 kg. After 2 months or 3 months, we will be doubling our body weight. Like 2 kg will become 4 kg. How the extra 2 kg came from? Do we add extra weight and stick to the body? No. From already cells are present. No, that cells will double and become 4 kg. But it is contrast with the non-living. For example, 1 kg flour is present. If you keep for 2 months, it won't become double. 4 kgs. What we are adding there? Again, we are adding 2 kg extra flour. Okay, that is the difference between the living things and the non-living things. So, this mitosis. And also another one scientist, he said, what is that? Rudolf Virchow. He proposed what is that? Omnicellular e-cellular. Okay, omnicellular e-cellular. Next topic we have studied about what? Cell cycle. In that cell cycle, we have studied what is that divided into 2 phases. This cell cycle is divided into 2 phases. What is that? Interphase. Interphase and as well as mitosis. So, interphase. Just I am repeating again and again this day because it has been a long gap to our NEET class. That's why I'm giving a glance upon the important points. Okay. So, interphase means period between. So, period between one mitosis and another mitosis and second mitosis. This is the period. Okay, this is the period between one mitosis and the another mitosis. And this interphase, okay, this interphase, it is divided into 3 phases. What is that? This interphase is divided into again 3 phases. G1 phase, S phase, G2 phase. Okay, and this interphase, it is totally related with nucleus. Nucleus. Okay, you have studied no already. I have made a pie diagram and shown you. Okay, I have made a pie diagram and shown you, right? So what is that? Period between one mitosis and 2 mitosis. In this, in which part of the cell is actively functioning? Nucleus. Okay, which part it is functioning? Nucleus. So you will get a question. What is the during interphase? Which part of the cell active? Which part of the cell is active? Nucleus will be active. So what is happening in the nucleus? After G1 phase, S phase, G2 phase. Interphase is divided into G1 phase, S phase, S2, G2 phase. So G1 means replication of chromosome starts. What is starting? Replication of chromosome starts. Okay, what is happening here? Replication is happening. Means what? One chromosome is starting to produce extra copy. Starting. Okay. So for example, one chalk piece is there. No, it is starting to produce second chalk piece. Okay, like that. One chromosome in a cell. So for each living organism, fixed number of chromosomes are present. For example, in human cell, 23 pairs are present. So each pair will start to produce extra copy. We will call it as duplicate or replica. Duplicate means what? It is same as original. It is same as original. Replica means what? It is same as original. So that is why replication. So here it starts. And here what is happening? It is synthesized totally in the S phase. What is that? Total number of chromosomes are synthesized. And G2 it is the stopping of replication. Here started here. In G1 phase what started? They have started. Okay, 23 pairs started to produce 46 pairs. Synthesis up to this phase, 46 pairs have been completed to be produced. G2 phase, it will stop. In this phase, what is stopping? One pair, one original, one duplicate. One original, one duplicate. So total, how many? So in our women cell, 46 pairs of chromosomes are produced at the end of the synthesis. And G2 phase, they will divide and they will stop. No need. So this is happening in the interphase. Please make a chart, write it in your notes again. And this G1, it is 10.5 hours. Okay, 10.5 hours. So I will write here. So how much time for G1? 10.5 hours. Okay, next. Synthesis phase. It is also 10.5 hours. It is 3.5 hours. So after the complete formation of chromosomes, no, at the G2 phase, they will get a signal to stop. So for stopping the duplication, it takes what? How much? 3.5 hours. So what signals will come? No need. Already you have produced an actual duplicate of the chromosomes. So you have to stop. For stopping only, it is taking how much? 3.5 hours. Now the cell. Okay, before interphase, how many pairs are present? 20. At this place, 23 pairs are present. At this place, how much? 46 pairs are present. So, for example, nucleus, no, where chromosomes are present, nucleus, inside the nucleus. So, the nucleus will be only fit to 23 pairs. Now, the 23 pairs has produced again 23 pairs. The space is very small, no? So that is why what the nucleus will do? It will slowly become bigger in size. The nucleus will become slowly bigger in the size. So what is happening after interphase? The cell will enter into mitosis. Okay, please write down. Then we will go to mitosis. Already we have completed mitosis. But what are the main points? Again, I am repeating because it is very, very important and very hard to understand also. Okay, whenever you listen, it will be very, like starting on. Okay, whenever you revise, no, at that time you feel like say new topic. Okay, every time when you are listening, you are thinking that it is a new topic. So again, I am repeating. So from one mitosis to another mitosis. So one cell is produced, two cells. So that two cells again they are producing four cells. So the time period between first one mitosis and another means continuous mitosis. That period it is known as interphase. Inter means no middle. Phase means stage. Interphase. This interphase means what? One mitosis, again mitosis. That is known as interphase. For example, first mitosis, second mitosis, third mitosis, fourth mitosis, like the continuation, no? So what is interphase? This is the interphase. This what is happening in this gap? That is known as what? Interphase. That is known as interphase. What is happening in the interphase? Nucleus is changing. The changes are happening in the nucleus. What type of changes are happening? Inside the nucleus, chromosomes number is changing. How much it is changing? Original number, it is getting double by the formation of duplicate chromosome. So that is divided into three phases. Start, completion, stop. So you can remember like that. It is start. Last one is stop. Middle one is total synthesis. That is why if you observe the phases names also. G1 phase, G2 phase. Because here start, here stop. In between, what is happening? Total synthesis. So at first, before G1, how many pairs? 23 pairs. After G2, how many pairs? 46. Okay. So from interface, the cell entered into mitosis. From which stage? From interface, the cell entered now into mitosis. So in the mitosis, two divisions takes place. One is related with the nucleus, and another one is related with the cell membrane. First one and second. Okay. Mitosis takes place with the two divisions. It undergoes two types of divisions. Okay. One is nucleus division and another one is cell membrane division. So nucleus will divide and cell membrane will divide. The nucleus division is known as karyokinesis. And the cell membrane division is known as cytokinesis. Cyto means cell. Kinesis means division. Karyo means nucleus. Kinesis means division. Okay. Nucleus and it is known as what? Division. So mitosis takes place with the majorly undergoes two divisions. Karyokinesis, cytokinesis. In karyokinesis, okay. So what is karyokinesis? First stage is metaphase. Metaphase. So what is the first stage in the karyokinesis? Metaphase. So we can say that mitosis has four stages. Okay, mitosis has four stages. But how many types of divisions are happening? Only at two places divisions are happening. Okay, you all members know that cell has many parts, no? Cell membrane. Okay, what is that? Cytoplasm, nucleus, mitochondria, vacuole, Golgi complex, endoplasmic reticulum, ribosomes, glyoxysomes, vacuoles, like that. Many are present. Chloroplast. Okay, so but every cell organ is not undergoing any change except two. What are the two? Nucleus and cell membrane. So nucleus, it is known as what division? It is known as karyokinesis. Cell membrane division is known as cytokinesis. So here in the four stages, first one is the metaphase. First one is the metaphase. So here you think in the before interface, for example, human cell. If we take human cell, okay. For example, here n. For example, n number of chromosomes are present at the starting of interface. When the cell is entering into the mitosis, it is getting what I said, no? One set of chromosome is producing other set, which is duplicate. So n will become what? N plus n, 2n. N plus n, 2n. What is here? N means number of chromosomes. So where chromosomes are present? In the nucleus. Where chromosomes are present? In the nucleus. What is the function of chromosomes? It carries the genetic information from parent to what is that? Progeny. We call as daughter cells. We will call. So chromosomes. So chromosomes are present in the nucleus. So now N has become 2N. Okay. So what is happening in the metaphase? First stage of mitosis is metaphase. So what is happening? Double size of nucleus increases. Size of nucleus, what is happening? Increases. And you have studied. What you have studied? This is the, for example, this is the nucleus. Nucleus is the opaque body which is present almost center in the, what is that? Cells. Okay, inside the nucleus you have studied. What you have studied in the general syllabus? Thread-like structures are present. What is that? Thread-like structures are present. So they are scattered here and there. But at this place, what is happening? They are getting condensed. Okay, what is happening in the metaphase? Their size of the nucleus increasing and the chromosomes are getting condensed. Condensing means what? Getting close. Condensed means what? They are getting close. Okay, so initial condition of chromosomes are they are thread-like structures scattered here and there. But in the metaphase, what they are happening? Condensed. What is happening? Condensed. And what is so in all this is the nucleus and this one is what? Nuclear membrane. Nuclear membrane. So another point is that nuclear membrane slowly disappears. So what is happening in the metaphase stage? Size of nucleus increases, chromosomes they became condensed, and nuclear membrane it is getting disappeared. Sorry, this is prophase. Mitosis like metaphase. This is prophase. Sorry. And next. And the centrosome appears. Centrosome appears. And after prophase, second one. Second, this is A. And next one is that metaphase. And next one what? Metaphase. Metaphase. So here spindle equator is produced. What is produced? Spindle equator is produced. And chromosomes are paired to one end. Okay, to one end. Both ends, they are going to both ends. Chromosomes are produced. So two sets are present, not two ends. Two sets are present. In that one original, one duplicate. For example, this is the pair of the chromosomes. Okay, it has produced what? It is produced another set of chromosomes. So what is happening there? It will be divided like this. Okay, that will be divided. So here spindle equator is formed in the second stage. Okay. Right. So I'm rubbing here protease. So anaphase. So here, first of all, the chromosomes will be like this stage. Okay. Now the chromosome is the second stage. They begin like this. Okay. So these chromosomes, this is known as centromere. Centromere. This is one chromosome. This is centromere. And this one, this side and this side, these are known as chromatids. Chromatids. So during anaphase stage, this chromatids shape will be appear like VLIJ. VLIJ. For example, V. Like this they appear. What is the shape of this? V. And next, L. Okay. This is centromere. This is chromatid. V shape, L shape. Next, I. Okay, I. J shape. So you will be asked one question. What is that? Different shapes are visible in which stage of mitosis? That is what? Anaphase. V L I J. So here what is this here? Centromere. So here how many chromatids are present? Two. But they are one set is to one pole and another set is dragged to another pole. So at that time the sets while they are moving, no, while they are moving, you will see different shapes. While they are moving, you can see different shapes. They appear like V, L, I, J. In which stage? Anaphase. Okay. So draw the diagrams that they are not given in your textbooks. So when you can see the shapes of V, L, I, J of chromatids, when they are moving, going to the corners with the help of spindle fibers, they appear like different shapes. Those are V, L, I, J. V, L, I, J. Next, last stage is telophase. Last phase is telophase. So what is happening in the telophase? Means, so at both ends the chromosomes have been moved. For example, this is the cell. Okay. So it has been formed here. So spindle fibers have drawn. Okay. Like this, many chromatids are formed to one corner and another corner. So what is happening around this around this chromatids? Around this chromatids, what is forming? The reappearance of nuclear membrane. So you are not seeing properly. So the nuclear membrane starts to reappear around the corners. So after that, it is forming through nuclear membrane. Nuclear membrane and nuclear membrane. So division of nucleus happens. So divided, no? Nucleus one part and another part. Up to here 2n. So now here 1n, one side to one side and one side to other side. After set has moved, nuclear membrane is again reappearing. So after here it projects like this and divides totally with one cell with one n chromosomes, one set of chromosomes. So now the chromatids, they will form how? Chromosomes. So what are the things happening in the metaphase, prophase? So reverse of prophase happens in the telophase. There what has happened? Cell size increased. Here cell size decreased, no, because two cells formed. There what is happened? Nuclear membrane disappeared. Here nuclear membrane appeared. There what happened? Chromosomes condensed. Here what happened? Chromosomes, they have been separated. Okay, they're called, here they are separated. So what are the points you have mentioned in the prophase? Contrast to the prophase, telophase happens. Telophase happens. Okay, this is the thing of telophase. Okay, so how many at the end of mitosis, telophase, how many has happened? One cell has formed into two cells. So how many sets of chromosomes are present in each cell? How much are present in the mother cell? That many are present in the daughters. Okay, at the end of the mitosis. Means what? Number of chromosomes is fixed. Number of chromosomes is fixed. So maintaining by mitosis, what is happening? We are maintaining the correct number of chromosomes. No change in that. Okay. So please write down. Then we will go with the some points. So division of nucleus happened, and at the end of the telophase, what is happened? Cell division, cytokinesis. Cytokinesis. So, mitosis was first discovered by the scientist Walter Fleming in which cells? Animal cells, and Strasburger in the plant cells in which year? 1879. So, Fleming was the scientist. He said about chromosomes. It was not given in your manual book. So, Walter Fleming was the first. He said that inside the nucleus, which are getting divided, not nucleus is getting double. Inside what is getting double actually? Because of the chromosomes getting double only, nucleus is becoming big size. So inside the nucleus, what is playing main role again? Chromosomes. Okay. If a cell wants to divide, which one is actively participating? Nucleus. In the nucleus, which one is actively participating? Chromosomes. So that was said by who? Walter Fleming. Walter Fleming. So mitosis you have studied, no? One cell it is becoming big and dividing. So that is why mitosis it is also known as, what is that? Division. What is that? Doubling. Doubling division. Doubling division. Okay. And this mitosis takes place only in vegetative cells. Vegetative cells. So it is known as somatic division. So mitosis it is known as what? It takes place in the what is that? Vegetative cells. So somatic division. Somatic division. Next one. It is also known as, from the very first topic of cell cycle we are discussing that. So we are becoming what? One cell it is becoming two. So that is why what is happening? Cell we are growing because of cell dividing. One cell is coming from other cell. So 2 cells, 4 cells, 8, 16 like that it is going, no? So that because of mitosis only we are growing. That is why mitosis it is also known as growth division. Growth division. So some 3 points we are here to discuss. One cell. So you can remember, one cell it is dividing into 2. One cell it is dividing into 2. So can you, by seeing this figure you will get some into mind. What is that? Amoeba, paramecium, euglena. One cell it is dividing into 2. Binary fission, no? So mitosis. So binary fission it is nothing but mitosis. So here in unicellular organism, so what we can say? Mitosis is helping for the unicellular organism for which purpose? For reproduction. For reproduction. But we are multicellular. Which is helping for reproduction means we can say that meiosis. Which division? Meiosis. Meiosis takes place in the sex cells. But here mitosis is taking place in the vegetative cells. But in unicellular organism, mitosis is helping for the reproduction. Mitosis is helping for the reproduction. And you also know about a technique in the plants. What is that? Grafting. Grafting, you know, for example, 2 plants. We have in my home, I have a guava plant, which is giving small guava fruits with sweet in taste. In your home, for example, in my neighbor home, they have big guava fruit yielding plant, but it does not have taste. So I need big fruits with sweet in taste. So what should I do? I should make 2 hybridization. I have to make. So what I am doing? I am taking my guava tree and cutting and taking the stem of the guava tree and keeping on the stem of the previous one. So at that time, what is happening? The cells of this cell and that plant will divide. Okay, so it helps for the grafting technique also. So mitosis it is helping for the grafting technique also. Next, during any work, our hand gets healed, okay, cut off, or we will get wounds. At the time, what is happening after many days? What is happening? The scar will be produced. Okay, wound will be healed. That is because of what? Mitosis division. Because the cells which are present above our finger, they become active and they divide. Before any wound happened, they won't divide. Whenever a wound is happened, the above cells will actively participate and they will divide. And also we can say that at a certain point, the cells, okay, they won't divide. For example, RBC cells are present. They will continuously divide. After 120 days, one RBC will give to another RBC. But the cells which are present on the skin, they won't divide except when our skin gets healed. Once you think if we go on for our whole lifetime, if we go on dividing, we will reach to the height of sky, isn't it? But to some extent, the mitosis is stopped. Whenever it is needed, the mitosis gets started. So this is about mitosis. So introduction, explanation, and again revision, and some more points we have. What is that? We have discussed again and again. Okay, facing on that points only. So next in the tomorrow session, we will go with the meiosis. Very hard topic we can say, because mitosis, it is doubling of the mitosis. It only takes place in only one gamete cells only. Mitosis will take place in many cells, whereas meiosis takes place only one type of cells, but it's very hard. It takes much time to complete also. Please attend the classes regularly. Tomorrow we will go with the meiosis. Write down the notes.